All right. This is the walkthrough video for Tuesday. It is September 4th in Try One Week 5. And we are indeed doing a little bit of bell work. So there's an assignment. I'm going to switch it so it's named 2.1. We're doing all the same things in Unit 2 that we did last year, but like the planning document is better organized at the national level. So it's going to be 2.1 or 2.1 and 2.2, but the name of the assignment is Malfunctioning Cellular Components. So if you go to Gradebook and you look for an assignment of this name, there's a little comment, even though you don't have a grade yet. And in that comment, I told you the name of your assigned condition for this project. So you're going to go find that comment, and that's what you're going to write down. My assigned medical condition is, and you're going to write down the name of that thing. So there's a little assignment where at some point in time next week, you are going to tell me and check your idea so that I can check your work and point you in the right direction. But you'll see if you go to that link underneath that is an assignment that's going to be due in about a week and a half. So I think it's due the 16th or 17th, the 17th. Oh, it's up here. It's right here. I'm looking at it. So it's due when you walk into school on September 17th. You're going to put two pieces of paper side by side, and that's how you're going to be able to make the poster that's described there. The references go on the back of the poster. The rubric on this assignment says it's worth 50 points, but it's actually going to go in as 20, the same amount as a unit note document. It's kind of a big thing. And so it's a cool thing to reward you for doing this like very professionalized thing that we'll talk more about soon. So does max out at 7 out of 10 points. If you would like to turn it in early, obviously you're welcome to do so. That's September 17th in 13 days. You can write same because today that's what we are doing. Welcome to Unit 2. So I just told you about the malfunctioning cell parts assignment. I also want to tell you about the video notes. The video notes for Unit 2 include an assignment that is strongly suggested but not required, and it is to draw a cell. We're going to learn about cell parts. And make sure you know how to draw where the things are in prokaryotic and eukaryotic cells. You might want to do that directly on your study folder, which you can start anytime. You can just grab a folder from me on that second shelf of school supplies that I'm pointing to. Today I'm going to do an intro to descriptive statistics. So I'll tell you a little bit about statistics. You'll get a paper that summarizes this tomorrow. And then we're going to do day 2 of exploring carbohydrates with bananas. Now, the things we're talking about today, are they on the Unit 1 exam? Your Dr. Milks is going to say that if they are, I'm going to tell you the answers during the test. That is my boundary. I did not express it very clearly yesterday, but today I'm going to. I will help you. I will teach you about how to do it during the test so that you have that capacity. The Unit 1 exam is scheduled for Tuesday, the 9th, and the Unit 2 exam is likely on the 30th. Here are Unit 2 guiding questions, and we're really thinking about living systems and their subcellular components as we think about this business of cells in bananas. Today, we're going to talk about chi-squared. This is the closest that Google has in its presets to that salmon color. We talked about chi-squared. Did I say chi-squared? We're talking about stats. We talked about chi-squared in the past, and today we're going to talk about measurements called standard deviation and standard error of the mean. As students who are in school today know, if you have not yet taken stats, you might leave today still in the middle of your understanding, still not really fully having a grasp on what's happening. That's okay. I anticipate that and expect that. You're going to draw a lot of bar graphs with a lot of standard deviation and standard error. You'll have to calculate it maybe 2 or 3 times, including today, but mostly you'll be given data and have to know what to do with it, which you'll know very clearly. So my fairy godfather, whose name is Brad, went out to this garage at his house, and it's like half of it is in shade all the time, and half of it is in the sun because of a tree that's in his yard in Kansas. And he picked ivy leaves to try to compare ivy leaf width under shady and sunny conditions. And so what you're looking at here is the average, right? Looks like it's about 7.5 centimeters for shady and about 6 centimeters, a little under that for sunny. But like, how sure are you that he didn't just accidentally pick the shady leaves that were bigger and the sunny leaves that were smaller? What if it's just random chance, just like we were thinking about in our behavior experiment with the fruit flies? Like, how do you know that that difference is real and actually like is something that happens when you change that independent variable of sunlight availability? And so the answer is these little bars that look like eyes. So this is some average. The eyes give us insight into how the experiment was conducted. So we're going to talk about that. Frank drew these leopards during the pandemic, and this is a screenshot of this picture, and he's so embarrassed by it that I'm going to show it every year forever. So he drew some wild cats. Don't worry about the fact that their eyeballs and faces are weird. Look at the number of spots. These leopards have the same average number of spots, but can you tell that the distribution is different between animals in the red population and the blue population? In fact, I can show you, we sample many, many of them. I can show you, if you sample a lot of these animals, the red population, that is a pretty narrow band. Sometimes they have a couple spots fewer, sometimes they have a couple spots more, but really they're about 100 spots on average. The blue is also 100 spots on average, but there are cats with very few numbers of spots and cats with 230 spots, right? That data is different. And you might do some different things based on this data. If you were, for example, designing a camera blind so that predators of this animal would attack it or something like this. Sometimes this knowledge really matters, right? The standard deviation is a number we calculate that helps us think about how spread out the data are. I'll show you that equation in a moment, but for right now, standard deviation is 10 in the red and 50 for the blue population. Here, we're looking not just at standard deviation, but standard error. And we're showing standard error, which is related to standard deviation, through these error bars that are up at the top of the bars. So what is standard deviation? It's a mathematical calculation that lets you imagine how spread out your data are. This is a normal distribution. Lots of things we measure in biology, if we measured enough of them, would end up in a normal distribution. In the lab, we will not be sampling 1,000 bananas, for example, but you could, and then you might get a graph that looks something like this, right? So when we calculate one standard deviation, we're doing a mathematical calculation that says, okay, if the data we were looking at had a normal distribution, what percentage of the data would fall inside that number? And it's defined as 68.3 if you take the average, the mean, and you go up one of these standard deviations and down one of these standard deviations. And so these error bars have something else in them as well. Because it's standard error of the mean, we've divided by the number of times the experiment was done, right? So if you got these averages, but you counted 10 leaves apiece, that would make you less sure of the average than if you counted 1,000 leaves, right? And you got that average. So these particular bars help us think about that. Now, we are right at the edge of my Star Wars knowledge. Do you know about TIE fighters? TIE fighters are twin ion engine fake starfighters from a fake sci-fi franchise, but you get the idea, right? There's a cockpit here, and then there are these wings, and the cockpit shoots out fake lasers. So exciting. And then what we're going to do together is think about how we can imagine these error bars as TIE fighters. Okay, imagine that these fighters are going to fly straight across the graph at each other. And it turns out that if they crash, like if they overlapped like this, like the top of the I bar hit the bottom, that would tell you that it's probably random chance that is causing the difference, and there's not a statistical significance between the two. But in this case, they don't. They fly past each other. And so there may be a statistically significant difference between the two. If you take stats, next we do a t-test. In APBio, we don't do that. This is kind of where we end. So I'm going to ask you to copy the sentences in this yellow rectangle directly into your lab notebook for this lab. They say, when the range of the error bars overlaps for 2SE, which we'll talk about how to calculate that in a moment, this indicates there's not a significant difference in averages and data sets. If the range of bars does not overlap, there may be a significant difference. So pause the video, make sure that goes under the banana lab, even if you haven't been here. Okay, so the original instructions ask you to count 5 cells in your field of view and count the plastids inside. We now know you need more banana to be able to do this well and to get enough in your field of view that this is easy. So here's my suggestion for you. Instead of counting 5 cells, you can count 1 cell and you count 4 separate cells. Also, I have sample data for you that I'll show you in a moment. So how do you calculate standard deviation? There's an equation on your sheet. If you know how to type it in your calculator, you can. But on the AP exam, you'll always be given that information. So for now, I just Googled calculate standard deviation. I found a calculator.net, seems very official. And I found a standard deviation calculator. You can figure out whether it's a population or sample. This is just a screenshot. And then you'll be able to calculate the standard deviation. Once you calculate the standard deviation, the screenshot is terrible. Once you screenshot the standard deviation, you're going to divide by the square root of the number of times you counted when you took your average. So like, what's the number you divided by when you were making the average? That goes here as n. Now, confusingly, it's going to be 4. And then as you'll see, when we make our graph, we have to multiply it by 2. So like, just do the algorithm for this time, just this one time for me. Okay, Drew Newsenwander is taking APBio in Mitchell, and he and my teacher friend Robin sent up these pictures. So this is a 400 magnification, right? So these starch in these plastids are made visible because of the iodine stain, right? And so he took 3 different pictures. This is a very unripe banana. This is a moderately unripe banana. And this is a ready-to-eat banana. They didn't have the banana of doom, which I'm definitely including next year when I make my sample data. So I've made these kind of bigger for you, as big as they can get, these pictures. And your job is to try to count out 4 cells. Ideally, you would try to find 5 cells, like maybe these 5 here you would try to count. Maybe you would count these 5, and like these 5. You would try to count them, and you would try to do that again here. Please notice that each one of these is a cell that doesn't have any plastids in it anymore. So do your best. It's like mini golf, and play it as it lies if you don't have your own data. So the next thing you're going to do is report in the standard deviation, the standard error of the mean. And then I want you to multiply that number by 2, which, yes, to do standard error, we had 4 counts, so you divided by 2. But humor me, 2 SEM. And then I want to tell you about 2 SEM error bars. So let's say you're going to get a piece of graph paper, and you're going to hold it like portrait mode. And let's say that this is what you end up with. This is probably not what your bars look like, but they might. So let's say that you have to label your axes here with a title, and you need to label your things here. But let's say that you have this bar and you made it the average and it's this tall. And for this particular data set, you calculated the standard error of the mean, which is also sometimes written like this, and it's equal to 1. To make the type of error bars that let you do that tie fighting nonsense, you are going to go up 2 and you're going to go down 2. And that's where the ends of the wings are on the error bars on those I bars. So more on graphing tomorrow after we finish the FRQ. Exciting times. We are going to pick this up, like I said, tomorrow. In the meantime, I wanted to remind you that because the unit exam is coming up, next week your study folders are due, which you'll turn in the day of the test. And there's a background assignment where you're going to do some multiple choice. You should check Canvas for that. The assignment deliverable is a piece of binder paper that goes into the inbox. Haven't done these before. We'll do all 8 of them this year. It is the last problem set to help get you ready for taking the test. You might have questions about taking those multiple choice. I want you to try it with your best brain in that best moment. And then if you get it wrong, you can learn from it. You're going to have to do 6 test corrections no matter how well you do. So, all right, that is it for me. Whoa, what is happening? Zoom. That's it for me. Hope you're doing well out there. See you soon. Bye. Cannot use the computer.